Well, hey, folks, welcome back. Now we're going to look at transport, stuff moving in and out of cells. Let me back this up and pull the cell up again so you can watch the animation. Oh, okay, it didn't work. Anyway, the idea is diffusion. Here we go. So with diffusion, substances move from areas of higher concentration to lower concentration until they reach a state of dynamic equilibrium. So with diffusion, there's this sort of random movement of materials. So they're bouncing off of each other, whatever medium they're in, whatever other molecules may be in the cell. And as they bounce around, eventually they move down their concentration gradient. They spread out. Okay. Now, this is a passive process, which means it doesn't require any ATP. They just use their own kinetic energy. So imagine playing pool. If you start off when everything's racked or together and you break, if the pool balls kept moving, eventually they'd spread out until they're fairly evenly distributed throughout the pool table. Well, that's what's going on with a lot of molecules. If they can pass through membrane, they will, until they reach, again, the state of dynamic equilibrium. Now, dynamic equilibrium does not mean stuff stops moving through the membrane. What it means is that it moves across the membrane at an even rate, so there's no net change over time. So things are still moving around. It's just that it's evenly distributed throughout the system. So that's like taking a drop of food coloring and putting it in water. The molecules will spread out, bouncing off of each other, water molecules in the glass, until they're evenly distributed. Now, osmosis looks at specifically the diffusion of water. Since water is so relevant to life, osmosis looks particularly at what happens with water. Now, this is an interesting example. This is what's called it's called a U-tube. So this was the U-tube that I was familiar with long before any sort of internet version of it. But with a U-tube, you can put a semi-permeable membrane, and then what they did was put these solute molecules, the little green sugary molecules there. Now, if you look at that, where it zooms in, the solute molecules, the sugars, are much too large to fit through the membranes, but the little water molecules can. So think about what happens in the U-tube. You have the right arm and then the left arm of the U-tube. In the right arm, there's a higher concentration of solute, right? So since there's a higher concentration of solute, there's a lower water potential. And water flows from high to low potential. So there will be a net movement of water molecules. I hope that was going the right direction in terms of what you guys are looking at. There's a net movement of water molecules to the right arm of the U-tube. So if you look in the sort of after image there, the second image, you can see how water flowed in until it reached a state of dynamic equilibrium, and the water actually moves up against gravity. So water can actually move against gravity as a result of water potential. Now, again, this is a dynamic scenario, so the water molecules are going to be moving back and forth across the membrane. It's just that there will be a net movement in one direction or another until it reaches that state of equilibrium. All right, so now let's talk about tonicity. This is stuff I'm sure you covered freshman year. Again, it looks at how cells will gain or lose water based on solute concentration. So there are two rules to remember. I would strongly encourage you to write these down and commit them to memory. It sort of makes it a foolproof way of figuring out what goes on. The first rule is that when we talk about hyper-hypoisotonic, we're describing the environment outside of cells. So again, generally speaking, unless you're told otherwise, when we reference hyper-hypoisotonic, we're describing what it's like outside of cells. The second thing is that water follows the solute. Wherever solute concentration is greater, there too shall go water. Now, unless there are some instances where water's flowing in and pressure potential goes up, but generally speaking, water follows the solute. So looking at the example of isotonic, iso means the same. I think like isosceles triangles all have something the same. Sides the same length, same angles, something. You know, everything's the same. Or two sides that are the same. Oh gosh, I'm sorry, it's been so long. But anyway, in an isotonic solution, you have the same solute concentration inside and outside the cell. Since the solute concentration is the same, what that means is that there will be no net flow of water into or out of the cell. Now, will water still enter and leave the cell? Absolutely. It does so at the same rate, so there's no change in water volume in the cell. One thing to mention, the little brackets, that means concentration. All right, hypertonic. Remember, we're describing the environment outside the cell. If you say it's hypertonic, hyper means above. Think of like hyperactivity, right? So hypertonic means there is a high solute concentration outside the cell. Well, hey, if there's a high solute concentration outside the cell, where's water going to go? It's going to go out of the cell. If water flows out, then the cell will shrivel up and plasmolyze or shrink. Okay. Hypotonic. Hypo means low. So think of like hypothermia or a hypodermic needle. Hypo is below, derm is skin. So a hypodermic needle will travel beneath the skin. Well, a hypotonic solution has a low solute concentration outside the cell. So that means solute concentration must be greater inside the cell. And since water follows a solute, water will flow in. Okay, so you're describing, unless you're told otherwise, you're describing the environment outside the cell, water follows a solute. So make sure you know that, because every year I've been involved with the AP exam, they have tonicity questions. This, again, is a nice image from the book about what can happen. A hypotonic solution, again, it's got a low solute concentration outside the cell, so water can flow in. And when that happens, it can cause the cells, an animal cell, to burst, because the increased pressure can make animal cells burst. That's why, don't put a goldfish in distilled water. Distilled water is hypotonic. Water would flow in, you'd have some fish problems. If you put a plant cell in a hypotonic solution, though, plant cells have those cellulose cell walls, so they can actually handle that pressure. So you can use like distilled water on a plant cell, and the plant would be fine. Now, isotonic solutions, that would be ideal for like our blood cells. That's why they'll use, if you get like an IV, they actually use a saline solution. So the saline solution is isotonic to our blood, so that you don't destroy your red blood cells. That'd be a bad scene. Now, in a plant with an isotonic solution, there's not much pressure on the cell wall, so the plant will actually be flaccid. It'll begin to lean over. Now, a hypertonic solution has a high solute concentration outside the cell, right? So if you have a high concentration of solute outside the cell, water potential is lower, water flows out. When that happens, the cell can actually shrivel up. And that's what can... I'm sorry, we've got an older dog, and he just passed gas. So I don't know if that was audible to you guys or not, but I apologize for him. So anyway, if you put a cell in a hypertonic solution, water is going to flow out and it can shrivel up. And with plasmolysis there, when things plasmolyze, you can get enough water flow out to actually pull enough water out to get the membrane, plasma membrane, to pull away from the cell wall, and that will kill the cell. So don't give your plants salt water. Not a good scenario there. Okay, when water flows in, we sort of talked about this. If you put a plant in a hypertonic solution, water will flow in. The water pushes on the cell walls, and it is turgid. Okay. When you have a plant in an isotonic solution, there's not much pressure on the wall, so the plant will go flaccid or limp. And again, plasmolysis happens if you put it in a hypertonic environment, because water will flow out, and the plasma membrane, the PM there, it pulls away from the cell wall. Okay, so that was all like diffusion, osmosis. Now let's turn our attention to facilitated diffusion. Note the name, facilitated diffusion. It's still diffusion. It's passive. You don't put any ATP into it. Substances are still moving from high to low concentration. It just gets some help. To facilitate means to aid or to help. So facilitated diffusion, it's still diffusion. It just uses the help of a channel protein or a carrier protein. Okay, so we have some different types of proteins that can aid facilitated diffusion. Gated ion channels, that's what actually helps you think. You can move ions to make action potentials to generate the electricity that helps create our thought. And then carrier proteins, again, they'll just change their shape and translocate the solute. So there you see a channel protein in the top image. The channel can be open all the time, or it can be gated and only open and close under certain conditions. The carrier protein, please notice that it will change its shape. And when it changes its shape, that translocates the solute. And again, in both instances, notice it's going from high concentration to low concentration. It's still diffusion. It's just facilitated diffusion. Diffusion with some help. All right, so now let's contrast passive transport with active transport. Active transport is active because you're using ATP. Okay, so active transport, you use ATP, and you actually push substances against their concentration gradient. You actually move substances from where they're less concentrated to where they're more concentrated. Okay, now, again, this happens to sort of create the conditions to allow for action potentials that allow you to think. The classic example of this is a sodium potassium ion pump. Those are ions, right? They're charged particles, so they can't pass through the membrane normally. So what happens is you use this pump to create a gradient. We'll talk about this more in the fourth unit when we talk about thought. But what happens is you use a protein, in this case a carrier protein. ATP will donate a phosphate group. Now, what do you think happens? If you put a phosphate on a molecule, what do you think it's going to do to its shape? That's right, it's going to change it. When we think of ATP, people always think ATP, energy. What's the energy, right? Energy is the capacity to do work, to make change. The energy in ATP is in its phosphate group that it hands off to molecules, and then it changes the shape of that molecule, and then it does something. So what ATP does is it locks onto that carrier protein. The carrier protein changes its shape, and then that translocates the solute against its concentration gradient. Okay. Now, if that's not clicking in your head yet, don't worry. We've got a series of images we're going to look at here. So let's notice what's going on. Here we're looking at the sodium potassium ion pump. So notice the sodium ions dock inside the carrier protein, and then here comes ATP. ATP hands off its phosphate and becomes ADP. ATP is adenosine triphosphate, right? It's got 3 phosphate groups, or 3 phosphates. It can hand off one of those phosphates to the carrier protein and become ADP. But look at what happens. When that phosphate docks, it changes its shape. The carrier protein changes its shape, and then it translocates the sodium ions. Now, notice where the sodium ions docked originally. They've changed their shape to help release the sodium ions. Now, what's interesting is now it can allow potassium ions to dock. And then when the phosphate is shed, the carrier protein reverts back to its original shape and dumps the potassium ions back inside the cell. So you continue that cycle. Sodium ions dock, ATP is added, the membrane changes its shape, translocates the sodium ions, and allows potassium ions to dock. That phosphate is shed, the protein reverts back to its original shape, and you start the cycle over again. Your capacity to think as you're listening to this and viewing this happens because these little pumps are working right now. So just using a protein that can change its shape, some ATP and some ions, allows you to perceive. Amazing. All right, so this is a nice summary of what we've talked about with transport. Passive transport, substances are going from high to low concentration without the use of ATP. Now, this can happen with just simple diffusion, if something is small, nonpolar, or you can have the diffusion of water through aquaporins. And aquaporins are a type of protein channel, which means technically it's a type of facilitated diffusion. It's diffusion, but with help. Protein channels are just like open tubes, and then carrier proteins change their shape. Now, in the case of active transport, the carrier proteins change their shape because an ATP adds a phosphate. And what it does is move substances against their concentration gradient. You move them from where they're less concentrated to where they're more concentrated, like the sodium potassium ion pumps. Now, let's look at what happens here. This is maybe looking ahead a bit, but when you move those ions in and out of the cell, you can actually create a gradient. What you do is you make the cell relatively positive on the outside and relatively negative on the inside, and that's referred to as a membrane potential. So by activating these pumps, by using ATP to run these pumps, you can redistribute ions and create potential energy, the capacity to do things. These pumps are running so much, a quarter of your calories just go to operating your brain. So, if you have a 2,000 calorie a day diet, about 500 of those calories just go to operating pumps in your brain. So you're putting a lot of energy into running this stuff. Let's see. This is what's referred to as an electrochemical gradient. So if you concentrate ions on one side of a membrane, you create not only a solute concentration gradient, you also create an electrical force. Okay. So there's sort of the chemical force element, which is about the ion concentration gradient. And then there is the again, difference in charge. So that's a membrane potential. So what happens is both the electrochemical gradient, or the electrochemical gradient, so the electrical force and the chemical force can drive those ions across the membrane, but only if there is a channel there. So that's like that red box. All right. So again, you can create these pumps like it's the sodium potassium ion pump. Excuse me. In prokaryotes, you can create proton pumps. And this is sort of interesting because the proton pumps, again, it creates this gradient that eventually was used in the evolution of mitochondria to power ATP production. So I think it's in the next unit we get into ATP production. So it's sort of amazing how that works. So here's what happens. This is just a teaser. So you use this pump to create this electrochemical gradient. So you have all these protons on one side of the membrane. And what the protons do is they have a concentration gradient and they have a charge gradient. It's more positive on the outside of that membrane. So what happens is when the protons flow back through this protein, looks like sort of a party there, but when the protons flow back through, it actually spins. It's a rotary protein. So it spins, and that causes the production of ATP. So this pump that was originally used in bacteria evolved to work with another protein to be able to generate lots and lots of ATP. So there's one reason you're alive right there. Another idea is what's called co-transport. Co means together, right? So what happens is you can create a gradient. So think about that proton pump. You can create a gradient. And when the solutes, in this case the protons, flow through another protein, it changes the shape of that co-transporter protein and it allows another substance to pass through. So here you see the proton pump creating the electrochemical gradient. The protons flow back in. When they do, it'll alter the shape of this co-transporter to allow sucrose, a disaccharide, to be transported inside the cell. Again, sucrose is way too big to fit in on its own. So it's going to need the help of this protein. Well, that can help you do that. That can help you concentrate those sugars. And you see this in what's called loading in plants. The xylem, those are the water tubes, right? The phloem is the tube that moves sugars through a plant. So I just think phloem for food. So this is how you can actually force sugars through a plant. Think of like cherries. Cherries are going to have a lot of sugar in them, right? Well, the sugar is made in the leaves of the plant. So if you have the leaves of the plant producing sugars and they pump it into the phloem, well, if you put a bunch of sugar in the phloem, what's going to happen to the solute concentration? Solute concentration is going to go up, right? Well, hey, if the solute concentration goes up, then water potential should go down. So what happens is water moves from the xylem into the phloem, where the water potential is lower, and then the increased pressure can drive the water carrying all that sugar to what's referred to as the sink. So then what you can do is pump these sugars into the fruits, and then that can concentrate sugars in the fruits. So it's a really cool way to drive the movement of sugars from leaves to the fruits just using water potential. All right, we'll stop there and you'll have one more video.